Dear students, welcome to the 7th session of Histology, Animal Behavior and Applied Zoology paper prescribed by the Mangalore University for 4th semester BSc Zoology. In this session, we shall consider the study of histological features of mammalian liver. The mammalian liver is composed of a single type of parenchymal cells called hepatocytes. The liver cells are called as hepatocytes. The liver is surrounded by a dense, irregular, collagenous connective tissue known as Glisson's capsule, which gives rise to septa that subdivide the liver into lobes and lobules. The liver produces bile juice and plasma proteins and has many other functions like storage of glycogen, detoxification of alcohol, killing of the pathogens that has entered into the body along with the food. The liver is enclosed in a thin connective tissue capsule which is irregular, dense collagenous connective tissue called Glisson's capsule. The liver receives a dual vascular supply. All the blood which passes through the intestine and the spleen is delivered to the liver by a special portal system called hepatic portal system and through the vein called hepatic portal vein. This portal blood carries not only nutrients but also various contaminants such as drugs, toxins from food, bacteria, and byproducts of blood cell recycling, which have been absorbed through the intestinal mucosa or produced in the spleen. You can see here the spleen has direct connection with the liver through the hepatic veins, the veins of the hepatic portal system, as well as the intestine has direct connection with the liver through the blood vessels. And this system of portal veins, where there is no involvement of heart, the blood from these organs directly flow towards the liver, where the liver performs the function of detoxifying these toxins and killing the harmful pathogenic bacteria that enters into the system through the food, as well as the blood cell recycling products such as the bilirubin and biliverdin pigments that are produced in the liver as a result of recycling of these byproducts of blood cell recycling that takes place in the spleen. The hepatic artery brings fresh oxygenated blood from the aorta. So this is what dual blood supply means. In one blood supply, the portal vein brings blood from spleen and the intestine towards the liver, and the hepatic artery brings the fresh oxygenated blood from the aorta. The portal venous blood from the intestine and spleen and arterial blood from the aorta mix together in hepatic sinusoids before leaving the liver in the hepatic vein. The basic structural component of the liver is the liver cells, which are called hepatocytes. Structural units of the liver are liver lobules. Liver lobule is hexagonal prisms in cross section and about 1 millimeter in width and 2 millimeter in breadth. Within each lobule, the hepatocytes are arranged into hepatic cards of 1 or 2 cells thick. You can see here, these are the structures which are called hepatic cards, where the hepatocytes are arranged like beads on a string, and they may be single cell or 2 cells thick in nature. Hepatic cards are radially disposed in liver lobules. See, this is the liver lobule, which is hexagonal in shape, and inside which we can see the hepatic cards that are radially disposed in the lobule. That means they will have a central vein from which they ramify on all sides. The cards of hepatocytes represent the parenchyma of the liver. So these hepatocyte cards represent the parenchymatous tissues of the liver. The space between the cards contains sinusoidal capillaries or liver sinusoids lined by fenestrated endothelium. So you can see here in this expanded picture of the hepatic cards. The hepatic cards are single cell thick layers of hepatocytes which are arranged like a beads on a string. And in between two such hepatic cards, there are some spaces, and this space will have the sinusoidal capillaries, and this space is called the liver sinusoids. And these liver sinusoids are lined by the endothelial cells. The endothelial cells means these are the cells of the blood vessels but are discontinuous in nature, and hence they are called fenestrated endothelium. So that means the endothelial cells possess many small pores in it, hence the name fenestrated endothelium. At the corners of the lobules, there are portal spaces which are occupied by portal triads. And the portal triads contains 3 important canals. See, these are the corners of the liver lobule. So look at this, the second picture. This is the diagram of the liver lobule, and in each corner of the liver lobule you can see three canals which are called portal triads. And this portal triads consists of interlobular vein, which is a branch of portal vein, interlobular artery, which is a branch of hepatic artery, and interlobular bile duct, which is the part of bile duct system. See, you can see here, there is an artery, there is a vein, and a bile duct. So a branch of portal vein will be there, a branch of portal artery, hepatic artery will be there, and a bile duct will also be there. So these three canals are present at each corner of the liver lobule and are called portal triads. The classic liver lobule is hexagonal in section. In the center of the liver lobule, there is a central vein. You can see here, this is a hexagonal shaped liver lobule, and at the center we can see a central vein. Here it is mentioned as CV. At the corners of the lobules, there are portal triads. We have already studied about portal triads. The blood flow is directed from periphery to the center. That means the blood flows from the peripheries to the center, and bile from the center to the periphery. So bile and the blood flows in liver through counter current mechanisms. The bile moves from the center to the periphery, while the blood moves from the periphery to the center. The portal lobule comprises of three adjoining parts of three classic lobules. It is triangular in shape and has at the center the portal triad and a central vein at the tip of each of its angles. The blood flow is directed from the center to the periphery and the bile from the periphery to the center. The hepatic acinus is situated in the adjacent areas of two hepatic lobules. The terminal branches of the portal vein, an arterial branch, and a bile ductule are in the center of the hepatic acinus. The blood flow is directed from the center to the periphery and the bile from the periphery to the center. The endothelial cells of the wall of sinusoids are separated from underlying hepatocytes by a narrow subendothelial space known as the perisinusoidal space or space of Dissay. You can see here, these are the hepatocytes. We call it as the liver cards or cards of hepatocytes. So in between two hepatocytes there is a space called sinusoid, which is lined by sinusoidal endothelial cells or fenestrated endothelial cells. And in between these two, in between the sinusoidal endothelial cells and the hepatocytic cards, there is a small space, and this space is called perisinusoidal space or space of Dissay. It contains the blood plasma. Microvilli of the hepatocytes extend into this space, allowing proteins and other plasma components from the sinusoids to be taken up by the hepatocytes. See, the hepatocytes possess some specialized structures called microvilli that extends into this space of Dissay, where the space of Dissay is composed of blood plasma, and all the components and proteins, hormones, and other chemical substances that are required for normal functioning of hepatocytes are present in the plasma of the space of Dissay, and through the microvilli the hepatocytes can absorb, take up these components for its normal functioning. So these cells of sinusoidal lining, which we call it as fenestrated endothelium, is composed of two more cells. The first type of cell is the endothelial cell, which is the cell of the lining of the blood vessel, blood capillary here. And second important cells are called macrophages. You can see here, these cells are called own Kupffer cells or simply Kupffer cells. These are found on the luminal surface of the endothelial cells, and these are typical macrophages. And I hope you know the function of macrophage is to engulf the pathogens that enter into the system. Hence, the Kupffer cells are the cells of immune system. Now let us look into the structure of an individual, a single hepatocyte. Hepatocytes are large polygonal cells. These cells may have one or two nuclei. Presence of a single nucleus is very common. Sometimes they may possess one extra nucleus also, but most of the time they possess a single nucleus. And the nucleus of the hepatocytes possess a prominent nucleoli. Some of the nucleus of the hepatocytes are polyploid in nature, and in most of the adults the hepatocytes are tetraploid in nature. That means they possess 4 sets of chromosomes. The hepatocytes have abundant endoplasmic reticulum. You can see here there are numerous endoplasmic reticulum, and there are two kinds of endoplasmic reticulums: smooth endoplasmic reticulum and rough endoplasmic reticulum. Both these smooth endoplasmic reticulum and rough endoplasmic reticulums are abundantly present in the hepatocytes. The rough endoplasmic reticulum forms aggregates in the cytoplasm. You can see here the rough endoplasmic reticulum forms aggregates. That means many rough endoplasmic reticulums are aggregated at one place, and several proteins are synthesized on the polyribosomes of this structure. We know that the function of rough endoplasmic reticulum is to synthesize proteins, and we know that hepatocytes have a lot of secretory functions, so they need to be always synthesizing some proteins that are required for normal metabolism. And it is the rough endoplasmic reticulum which are abundantly present in the hepatocytes perform these functions. The smooth endoplasmic reticulum, you can see here the smooth endoplasmic reticulum, and this is responsible for the process of oxidation, methylation, and conjugation required for the inactivation or detoxification of various substances that has entered into the system through the food. And each hepatocyte have approximately around 2000 mitochondria. You can see here everywhere the mitochondria is dispersed. Since the energy requirement of these hepatocytes is too high, so a lot of mitochondria are present in the cytoplasm of the hepatocytes. Hepatocytes do possess the suicidal bags of the cells that are called lysosomes. And the lysosomes of hepatocytes are responsible for degradation of intracellular organelles and endocytosis of many macromolecules. So along with these lysosomes, there are also some structures, vesicles called peroxisomes that are also present in quite high number in the cytoplasm of the hepatocytes. There are many Golgi complexes as well, and the function of these Golgi complexes is to produce lysosomes and secretion of the plasma proteins. So once the liver synthesizes and secrete the bile components, it must be transported to the required organ, which is the duodenum. Hence, this function is performed by a system of vessels called biliary system. The biliary system is the system of canals on which bile from a liver flows to the gallbladder. You can see here there are many biliary branches, canals. All of them come together to form a single duct called common hepatic duct. Okay, there is a left branch and the right branch. The right hepatic duct brings the bile from the right part of the liver, and the left hepatic duct brings the bile and other plasma proteins from the left part of the liver. Both of them coalesce, both of them merge, producing a common hepatic duct, and this bile secreted from the liver is stored in a structure called gallbladder, which is green in color here, and generally it is also green in normal conditions also. And then the bile flows to the duodenum. The biliary tract includes intrahepatic and extrahepatic passages as well. And then whenever there is requirement, the gallbladder releases the bile present in it and there is a common hepatic duct that reaches the pancreatic duct. So the pancreatic duct from the pancreas and the common hepatic duct from the gallbladder, both of them merge together near the duodenum, and their secretions are emptied to duodenum through a common duct called hepatopancreatic duct. This is all about the anatomy and histology of mammalian liver. And these are some of the textbooks that you can refer to study more about the anatomy and histology of the liver. Thank you.